there is a discrete species covered about 25,000 different species. Unfortunately, in the northeastern region, more particularly in this biodiversity hotspot region, no precise documentation of this insect biodiversity still has not done. But however, these indigenous people somehow, by utilizing these resources, maybe they're conserving, or maybe they're at the same time they're also there is no report that at the same time they might be using extensively also. These insect resources, among these insect resources, I've taken up this entomophagy. Because in this, while studying this entomophagy, that is under this entomophagy aspect, that not only I will cover the insect that are consumed as a food, and in studying this, it also has various broad aspects that people are consuming this insect not only for the nutrient supplement, but also for as a traditional medicinal treatment also, and at the same time it also enhance their economic input for their livelihood. So scientific validations of this traditional wisdom in biohospitaling will assume great significance. For this process, I have done survey work in different regions of the state, and the insect, whatever they have, the traditional people reported for any purpose of utilization, there are collected information, and live specimen is also collected and preserved, and I'm trying to build an insect resource museum also in my lab. The importance of this selecting this entomophagy is that it has been using since the Paleolithic era, and there are also many important other features that this insect in terms of biomass it is numerous, and in terms of reproductive capability it is very strong, and at the same time it also gives high nutritive value, and its existence is also very that is present in every ecosystem, and at the same time it is that natural renewable resources, and it also becomes a means to generate income for the living. Some of the examples we have.
Can you summarize please? Can you summarize in this moment?
Yes, sir. This is Oryctolagus rhinoceros is one of the pests that infects the different beans. And these in the edible form it consumed as a raw seed and the fat food, but at the same time this larvae they are using in the disease treatment that this get crossed and different infection by the applying the lipase. And about this maximum percentage of the orders that using insect resources is about 50% of the belongs to the Coleoptera species. And the fourth is entomophagy. The different biohospitaling aspect is that it can also give medicinal input and aesthetic input, and the industrial aspect is also there. At the same time, it can also be means for the biodiversity conservation. While considering the nutritive value, also is while comparing with the conventional food, that is in terms of energy yield, that is compared with the fish, there might be more than 3%. Next. As a conclusion, that this entomophagy is comparable with other conventional food products, and at the same time its significant importance is also nutritional, economic input as well as medicinal input is also there, and it may be formed as a new class of food which can render low input, small business and small commerce.
Thank you.
And organizer for giving this opportunity. So the time has come when we must consider for adopting a system by which the sustainable agriculture can be maintained along with increasing the yield. So the application of associated bacteria, which includes rhizosporic bacteria, rhizosporic bacteria, and endophytic bacteria, can be very efficient for attaining this goal in various manners. Endophytic bacteria has H over its rhizosporic counterpart or rhizogenic counterpart in main two regions, that it directly benefits the host plant by residing in the interior, and it has less competition in comparison to that of riboscale. So the can enhance the plant growth by attributing certain properties. The one is to nitrogen fixation and another phosphate solubilization. So it can be used as biofertilizer by fixing atmospheric nitrogen, which is not possible by the plants and other animals. Then the another aspects are phytohormone production. The bacteria are known to produce phytohormone like linoleic acid and some other phytohormones. So it can enhance the root development, particularly lateral root development. Condition like the stress condition, biotic or abiotic stress condition. Plants produces ethylene as a defense mechanism. At certain level, this ethylene is better for the plant growth, but when it exceeds, it can inhibit the plant growth. So some bacteria produces ACC deaminase, which degrades ACC, that is the precursor of ethylene, and thus enhances the root and shoot development. Siderophore production. So that siderophore production causes the filtration of iron compounds from the atmosphere and provides the iron to the plants. The other aspect of endothetic or associated bacteria is biocontrol. Biocontrol activity, it can inhibit the growth of pathogenic microorganisms and it can execute it through production. That siderophore production is also helpful because it deprives the pathogen from iron and thus helps the growth of plants. Then due to competition for nutrients, it can also inhibit the other microorganisms to grow in the rhizosphere or in the plants. Then through lytic enzyme production, it can inhibit the growth of many pathogenic organisms. And lastly, induced systemic resistance. So this induced systemic resistance refers to the ability of the plant to produce compounds that can inhibit pathogenic organisms. So this kind of bacteria induces the production of compounds that provides the plants a kind of immunity. Bacteria that were identified initially by biolog method and then by 16S ribosomal DNA analysis. So these were identified as actobacter substance into the bacterial species, Klebsiella, Klebsiella oxytocin, and Bacteria dispersa and Bacillus. The variation on molecular level was further estimated by using amplified ribosomal DNA restriction analysis with the use of two restriction enzymes, that is L1 and R7. The other molecular fingerprinting techniques were also used, which were RNA and STS-based for total protein profiling was also used for the same. We further characterize the diametrophy of these isolates because our aim was to screen for nitrogen-fixing bacteria. So in various conditions like various carbon sources, the nitrogen fixing ability was estimated, and it was found that different isolates showed varied response to different carbon sources. So it may relate to the suitable carbon sources which is necessary for the growth of particular bacteria. Then, as we know that nitrogenous activities or nitrogen enzyme is sensitive to oxygen concentration, that was also tested in the presence of aerobic and anaerobic conditions, and it was quite obvious that the nitrogen activity is inhibited at certain level of oxygen concentration. So found that after 2% of oxygen, the inhibition of nitrogenous activity was observed in almost all the isolates. Further, for confirming the expression of nitrogen is entered. So this is the system block. So merely isolating bacteria after surface sterilization does not ensure the endothetic nature. So to track the endothetic nature of these isolates, certain isolate was tracked with a molecular marker like dust state using transposon based plasmid, and then it was infected to the plants. After 21 days of inoculation, the bacteria was section, the plant groups for section and assigned for the gas production. And is there any enzyme, certain enzyme is there which is utilized by this bacteria for getting integrated and getting colonized in the plants. So we tested for certain cellular activity and bacteriolytic activities in these bacteria, and in not all but in majority of the bacteria, all these activities were found, particularly in the except for the effect of these bacteria on plant growth. So it is obvious from this result that it enhanced the plant growth up to 52% of the root increase. So it is concluded that this kind of bacteria can be used as a efficient biofertilizer. And
I was research for the DSP scientist project under Dr. Shardi Sundar, University of Allahabad. The registration susceptibility of the plant from the pathogenesis by mention of the plant program cell. I'm very happy to deliver my presentation and the friend of the Dr. Dhariwal, who is so interested in this type of work. This biotic and abiotic distress, the plant environmental development within the plant is changed. Due to the stress response in the plant tries to protect themselves against the different stress by operating the suitable signaling pathway, like calcium-mediated pathway and systemic acquired resistance pathway and the lismonic acid-mediated pathway and the salicylic acid pathway. During the plant-pathogen interaction, there is a plant and the parasite, like action, or there is a few of the possibilities, like defense response in the plant, show the defense against the plant and the protective response. If they are not protecting, then this defense response is not stimulated by the plant system, they show the disease, or they show other the symbiotic relation between the plant and the pathogen. During the plant-pathogen interaction, the pathogen basically find the host for take up the nutrition because they want the nutrition to grow and survive on the plant system. The successful pathogen must overcome the defense barrier of the plant system and evade or tolerate the resistant mechanism of the plant system. And the susceptibility of the infection basically how the plant system is defeated by the pathogen. Here we need the modern system is a canal disease of the bee. It's a very important economic disease of the plant system, and basically it is a seed pod disease of the bee. And this falls under Pinaceae indica. It is a semi-parasitic type of the fungus. It's an important fungus for the culinary prospect. The wide distribution of the India and also other parts of the world like US. And basically the annual loss by the disease is of 40,000. And the reduce the breed quality and also the quantity as well as because the KB is very important. Basically it is a seed pod and it is reduce the viability of the seed also. In this work I suggested two variety of the wheat. One is the AC29 and the second is the WS542. The AC29 is show the high resistant variety on the basis of the plant breeding. And also select the different stages of the developing wheat spice. For which is seed pod disease, we are select the different species of the stages of the wheat spice. And this is a particular different stages. We are basically infected of the plant. Food stage is not very prone for the disease. Basically, when the ear has just dipping out, the S1 stage. Just shown in the figure in section S1, it just shows the higher percentage disease compared to the other stages of the development. In this, we are discussing about the physiological and the biochemical stages. We are explaining the hormones in the developing wheat spice with the help of the analyzer. And this one is governed in the GKTK Bangalore. There is a facility they develop the polyclonal antibody against the hormones. And they are basically the cytokinin and the different types of the cytokinin like IPA, TSLR and the acetic acid. The level of the cytokinin is not so very significant result, but IPR is very high in the resistance and the susceptible TSLR is very high in the susceptible variety. But the whole of the ADA shows a very high in the resistant variety and very much S2 stage. This is very prone for as well as H2S state, they are very much prone for the disease. They are like the phenolphthalein pathway, like polyphenol oxidase and the peroxidase. This is very high in the resistant variety. And they are basically provide the structural and the mechanical defense, and with the help of the stimulation and lubrication of the cell wall, and provide the nutrition from the dead surface. And the prolonged bed rest is a very essential step for the growth of the semi-biotrophic type of the fungus. That on the further study, because the cysteine-48 inhibitor is very high in our resistant variety, and also the communication mediated pathways show the positive result. We are subsequently prone and characterized the cystatin gene family. For cystatin RNA imaging methods, we go through the RT-PCR, clone these gene and screen these gene and reduce the sequence. And basically in cystatin is a 5 gene family, cystatin 1, 2, 3, 4 and 5. In my case, only 3 genes are stimulated. And basically in the resistant variety, the very high show the WC1, 2 and 3 show very high results. And these are the semi-quantitative with the help of semi-quantitative RT-PCR, and also densitometry analysis, and also check with the help of the methodology. We are basically conclude that protease and the cysteine protease interaction basically govern the resistance and the susceptibility to the plant system. If the protease is rated is very high, provided the fungal hyper, it shows the susceptibility. If the plant show defense mechanism, they use the cysteine protease inhibitor against these proteases and suppress the growth of the semi-biotrophic type of the fungus like finish. Dr. Shanti Sundaram, Suprel, Ifrat, Soumya, Prashant, Ashutosh and Priyaka, and also I give my thanks to DSC, Department of Science and Technology, New Delhi, for providing me a young scientist project under fast technology.
Now, in the time, because otherwise the next session will also get delayed. So, anyway, I think we are not very late. Just 20 minutes behind the schedule, it is 10:50. So before I formally close this session, I request the session deputy at Dr. Seema to give a quick overview, quick summary of the presentation.
Thanks the proceedings of the session. We had interesting talks, starting with Professor C. Raghu Kumar, who stressed upon the importance of ecologically directed bioprospecting of marine fungi and their utilization in various industries, pulp and paper, distillery industry, dye industry for bio-bleaching, bio-pulping, even for the de-inking of the Xerox paper. The next talk focused on the strategies for combating anti-HIV drug resistance, and potential use of PNA as a chemotherapeutic agent was also discussed. Further talks highlights the importance of entomophagy and stressed on the preservation and utilization of insects as an important bioresource. The characterization of the putative endophytic diazotropic bacteria, which were isolated from various varieties of rice, was also discussed. Then the resistance